So good morning, and thank you for the introduction, Dr. Brem. Again, I'm Natasha Shaybani, but today I'll be presenting work that's predominantly been performed by an undergraduate student in our lab, Alec Batts. As the title of this talk suggests, we are interested in looking at extracellular vesicles, and these vesicles tend to fall in two major subsets, one being microvesicles, otherwise known as exosomes. These can be on the order of 200 to 1,000 nanometers and typically directly bud off of the plasma membrane. Meanwhile, there's a smaller subset of extracellular vesicles known as exosomes, and these typically tend to be on the order of 30 to 150 nanometers and are formed in a slightly different fashion whereby they're secreted through multivesicular endosomes. Taken together, the notion of extracellular vesicles has become very interesting lately because these are differential carriers of cytokines and growth factor receptors, RNAs, and other payloads that are capable of cell-to-cell signaling. So in the last 2 decades, there's been a resurgence of research in this area. And we were interested in exploring this in the context of focused ultrasound. So the aim of this study was to examine the impact of FUS on glioma-derived extracellular vesicle release and profile. And we essentially designed a study wherein we were able to assess this in vitro. First, we seeded U87Mg or GL261 Luke2 cells into acoustically transparent cell culture chambers. We subsequently exposed these cells to focused ultrasound, and I'll discuss the regimens momentarily. 15 minutes later, we harvested supernatant and clarified it for exosome isolation or extracellular vesicle isolation via differential ultracentrifugation. And finally, we characterized our EV populations via nanocyte to profile size and concentration profiles and mass spectrometry to garner information about the proteomic profile of these EVs. So the schematic here shows what our focused ultrasound system looked like. We essentially had an imaging transducer that was orthogonal to a focused ultrasound therapeutic transducer, and this was for the hyperthermia regimen specifically. We immersed the acoustically transparent cell culture chamber into degassed water and subsequently treated using various parameter sets that are detailed in the table. So I won't read those out other than to mention that our hyperthermia regimen achieved approximately 50% peak focal temperature based on our simulations. And our microbubbles were albumin shelled bubbles that were 2 to 3 microns in size. So based on our conjecture that FUS should sort of modulate extracellular vesicle release, we first were able to demonstrate that both thermal and mechanical FUS exposure elicit glioma-derived extracellular vesicle release. So on the figure to the left, we're looking at the hyperthermia regimen, wherein we observed that there's a significant increase in the concentration of the EVs that we isolated subsequent to focused ultrasound hyperthermia. And on the right, the figure demonstrates that at 2 different peak negative pressures that we assessed, 300 and 500 kilopascals, there was again a significant increase in the extracellular vesicle concentration derived from our glioma cells. Moreover, we didn't observe any major fluctuations in the size profile of these extracellular vesicles. Most everything that we isolated between the control and focused ultrasound groups retained an average diameter of about 100 nanometers. So next, we were interested in assessing whether FUS hyperthermia modulates proteomic profile of these glioma-derived extracellular vesicles. And our first pass results after mass spectrometry are promising. So here we're looking at a few different markers of interest. So I'll point your attention first to HSP 70 and 71, wherein we see that in our treated groups there is an increase in the expression of these markers, and we would sort of expect this with a hyperthermia regimen. Moreover, when we look at sort of some classical markers of extracellular vesicles, and more specifically exosomes, we observe that those are represented as well. So flotilin 1 and flotilin 2 seem to increase as a function of treatment. Finally, RAB1 is a marker of membrane uptake and exosome formation, and once again increases for now as a function of treatment. So Further, we did cryo-EM because we were sort of taken with the fact that we might be isolating exosomes more specifically. And our EM results not only seem to be consistent with the structure and morphology that we would expect of exosomes, but further recapitulated our size distributions that we found by nanosite that I showed in the earlier slide. So we're following up on these results now as we see a marked increase in glioma-derived EVs, as well as some potential impact on the proteomic profile. We'd like to dig a bit deeper and profile the EV subtypes to understand discretization of microvesicles versus exosomes in these culture models, as well as now begin to assess the differential impact of thermal and mechanical exposure to FUS more closely in this context. So with that said, I would like to thank everyone in our lab and all of our collaborators who have been instrumental to these studies, as well as our funding sources. Thank you.
Questions?
Very nice talk. Did I miss something, or did you not show the FUS data? The mechanical effects. Yeah.
For mass spec?
Yeah, mass spec.
So we haven't done it yet. Those samples are sitting in the freezer, and it's on the top of our priority list.
Hi.
Yes.
When you mentioned hyperthermia, because the cells are in suspension, how are you sure you're really heating the cells?
So these cells are actually adherent when we expose them to hyperthermia. We've been using the OptiCell or Pitaka platforms, so we actually seed the cells 24 to 48 hours prior to treatment and allow them to reach a certain confluence before exposing them to FUS.
Yeah, my question is, you apply parameters that, they're supposed to be generate hyperthermia, but because they're in suspension, you don't have absorption, so you aren't really heating some of the cells. You may only create radiation force and something.
Absolutely. So having seen this effect, these are some of the questions that have been on our minds. So we're now beginning between the combination of in vitro recapitulation of what we've been doing and simulations. We're trying to understand the extent to which we're actually heating cells versus plastic and anything beyond that. So it does become more complex when you think about the dynamics of heat transfer in this model. Yeah.
You planning to look at anything else besides a mass spec, like miRNA and?
Yes, and that was something I didn't explicitly mention, but our approach was to sort of take a broad sweep, but people have done lipidomics, they've of course done RNA-seq and other genetic profiling, so there is a broad array that in our arsenal that we can begin to unpack now.
Just a quick
Last
question.
Yeah.
Did you show any viability data on the cells that were exposed? Because if you put cells in suspension or even adherent in a tube with microbubbles and free fluid, the first thing you have to ask are you might be seeing release of the exosomes, but are the cells still remaining viable? Because that's not a realistic kind of scenario of how the cells would actually be treated when they're inside a body or inside a tumor.
Right, so for the hyperthermia studies, for now we've only done viability assays via trypan blue, and we don't see a significant cell death component associated with that treatment. For the mechanical, we're actually going back now to characterize that using the same cell culture platform that we established for the GL261 cells. So I completely agree. Again, that's a really important component, and we need to unpack that question more closely.
I want to consider putting them in a gel or something like that. So the cells
We've actually, it's funny, there's another student in our lab who's really interested in taking that particular approach, so it's very timely to hear that suggestion.
Thank you. Thank you very much.
Thank you.